Bien, por favor vayan tomando asiento, que vamos a iniciar la conferencia plenaria, por favor. Bien, muchas gracias. Comenzamos. Tiene la palabra la doctora Esther del Olmo.
Buenos días. Sí, ya. Good morning, ladies and gentlemen. It is my pleasure and honor to introduce you to the prestigious researcher, Professor Manuel Elkin Patarroyo, who will give today the plenary lecture titled The New Vaccines. Born in Ataco, Tolima, Colombia, he felt from childhood great admiration for the figure of Louis Pasteur because he read a comic based on the French scientists. He earned his PhD in medical sciences at the National University of Colombia in 1971. His great curiosity and interest in immunology and virology led him to make several postdoctoral stays at Yale and Rockefeller Universities in New York with Professor Kankal and at the Karolinska Institute, Stockholm, with Professor Klein. He has been full professor of the National University of Colombia in Bogotá, an associate professor at the Universities of Rockefeller in New York and at the University of Stockholm. In 1976, he founded the Institute of Immunology at the San Juan de Dios Hospital in Bogotá, which is dedicated to the development of synthetic vaccines, including the malaria vaccine against the terrible parasite Plasmodium falciparum. He developed the first synthetic vaccine produced chemically, the first against a parasite, and the first against malaria produced by Plasmodium falciparum, which was donated to the World Health Organization for the benefit of the humanity. The results of this first fully synthetic vaccine were published in Nature in 1987 and 1988, followed by a long series of clinical tests and field studies that were published in Lancet and other scientific journals of high global impact. Professor Patarroyo has received several national and international awards, including the Prize of Médecine de l'Année in France, the Léon Bernard Prize of the World Health Organization in Switzerland, the Robert Koch Prize in Germany, the Prize of Asturias Awards in Science and Technology in Spain, the Edinburgh Medal in England, the Prize of the Academy of Sciences of the Third World, and he has been awarded 29 doctor honoris causa from different universities in the world. He has published more than 400 articles in scientific journals of high international impact, such as chemical reviews, Nature, Lancet, accounts of the chemical research, and others. He also made contributions to 26 books. Since 2001, he is the founder and the current director of the Fundación Institute of Inmunology of Colombia, the FIDIC. Currently, Professor Patarroyo's research is oriented towards the search for a logical and rational methodology for the development of vaccines for the various infectious diseases that mainly affect people who live in tropical regions. In 2006, we have the honor of counting with his contribution for the inauguration of the Center of Tropical Diseases of the University of Salamanca, the CIETUS. Please welcome Professor Patarroyo, who will talk about the new vaccines.
Thank you very much indeed, Esther. It was very generous of your part, that, how will I say, generous presentation indeed. You know, I would like very much to express my gratitude to the organizing committee of this event, yeah? Especially to Antonio Muro, who is here with us, and his team, who have been really struggling and doing lots of efforts to bring excellent group of scientists and to share with you, obviously, the data that they and we have already published. Thank you very much indeed, Esther. It was very kind of you. And I would like very much to acknowledge also that I have been associated with this university for at least 20 years. And it was thanks to Ignacio Verdugo, who's in here, you know, by the time that he was the rector of this university. And with Antonio Muro for I don't remember how many years. He's getting old, definitely, you know, so that's the point. So thanks again, and thanks to all of you for staying and attending to this lecture, which I would like very much to share with you all the data that we have developed in the search and the purpose to develop a logical methodology, as Esther just mentioned, for vaccine development. Can I have this working on? Is that okay? So, what I would like very much to do, obviously, as I mentioned to you before, is to express my gratitude in here, yeah, for that invitation in the Innovation in Pharmacy, Advances and Perspectives, yeah, to talk about the new vaccines. What is the purpose of this one? This is our institute that Esther just mentioned, the Fundación Instituto de Inmunología de Colombia, who followed the Instituto de Inmunología del Hospital San Juan de Dios in Bogotá, Colombia. Unfortunately, that institute was embargoed by, please don't take it in a wrong way, by an Spaniard bank, you know. But anyhow, it was that the reason we had to found 16 years ago another institute, which is the Fundación Instituto de Inmunología de Colombia, which is associated with the Universidad Nacional de Colombia, Universidad del Rosario, Universidad de Ciencias Aplicadas, the university I do all the veterinarian trials, you know, and the Universidad de Boyacá. These are the universities that are now supporting our research in Colombia, yeah? And next, please. The purpose of our institute has been, for the last 40 years, 40 really, to identify the rules and principles, as Esther just mentioned, for a logical and rational vaccine development. Vaccines that have been developed so far are biological products, meaning, in other words, that according to that methodology, one takes the microbe, mutate it, or look for mutants that exist in nature to induce the defenses but not the disease. So you kill the microbe or you mutate the microbe. That's one of the methodologies for biological vaccines. The whole microbe used to immunize the person. But there are some other biologically derived methods, like, for example, recombinant molecules or vector-based vaccines, where you insert into a virus or into a different bacteria part of the genome of the disease that you want to prevent, or DNA vaccines. But all of them are biological products. Ours is, as Esther mentioned just before, just a few minutes ago, is the first chemically synthesized vaccines. And then what happened was that was as a consequence of our purpose to identify these methodological rules for chemically synthesized vaccines that allowed us to postulate this concept. The immune protection inducing protein structure concept is a new concept that has developed its own methodology, leading to the results that are the new vaccines. That's exactly the point. This is a consequence of my previous studies, as she mentioned before. I did my studies at Rockefeller University, you know, while doing my medical studies at the National University of Colombia with these two giants, Henry Kunkel and Jerry Edelman. Jerry was Henry's student, and both of them were as excellent immunologists, not only that, but also excellent chemists. So I learned protein chemistry from both of them, yeah? So much that, in essence, yeah, because of that, next please, yeah, because of that, I invited Henry several times to Colombia, and I published 15 papers with him, including my first paper in Nature that it was long time ago, yeah? But also, just across the hall, there was this other guy, who was Bruce Merrifield. He knew, and he was the one who developed the chemically synthesized peptides concept. So it was relatively simple for me just to learn protein chemistry, amino acid sequencing, and protein structure from Kunkel and Edelman just across the hall, Bruce Merrifield's methodology to do peptide synthesis. I asked them, I remember very well, when I was 20, do you really think that chemically synthesized vaccines are feasible? And then Bruce responded in a very sarcastic way, yes, but it will take you the whole life. And I said, well, it's my life and I can spoil it the way I want. True, that was the story, really. And it's true, so far I am 72, I have not finished so far, so that is my whole life, definitely, yeah. So that was the reason. This is former President Belisario Betancourt, a very good friend of Ignacio. And this is David Baltimore, who was just one floor above my laboratory, who was the one who discovered the retroviruses, you know, and among them, the AIDS virus is one of them, you know. So that this is David Baltimore, who got the Nobel when he was 38 years old. And this is Bruce. When he got the Nobel Prize, the announcement was made while he was in Colombia, and that was by October 1984. So I had to fly directly from Bogotá to New York to respond to all the questions, and that was Bruce. But my interest in malaria was as a consequence of this guy. Peter Perlman was a full professor and secretary of the Nobel Committee in Stockholm. And then he told me, hey, listen, why don't you work on malaria? And I said, well, I'm working on tuberculosis. And then he said, yes, but the experimental model is not the appropriate one. So I said, what? He said, yes, the experimental model for tuberculosis is the guinea pig. And even with the vaccine, the guinea pigs are infected. And I said, what? He said, yes. Why don't you work with malaria? And so that he was the one who convinced me to work on malaria. His son, Thomas, is nowadays the president of the Nobel Committee, you know. Bo is, let's say, is something like a heritage, something like that. So that was the reason I got involved in malaria, yeah. And I proposed by then, a long time ago, that a deep understanding at the atomic level of the malaria parasite molecules and the immune system molecules and their interactions are necessary to be understood for a logical and rational vaccine methodology. And that's what I want to share with you today. Yeah? So, malaria, why? Malaria because it's a very big public health problem all over around the world. There are around 200 to 300 million cases of malaria a year. A year, that's a lot of people. And also, there are around 700,000 deaths a year. As you well know, malaria is caused by the bite of this Anopheles mosquito, who, looking for blood, introduces the proboscis under the skin and leaves the parasite that she has in the salivary glands. And these are the larva-like structures that are called sporozoites. But let's call it larva-like, you know, because they look like a larva. And wherever they are deposited, they move at this speed. So usually each bite of the Anopheles mosquito inoculates something between 1,000 to 10,000 parasites. But let's take the lower number, 1,000. What it happens is that this parasite goes through the small vessels to the liver, and in the liver looks for a hepatic cell with a special amount of heparan sulfate proteoglycans, and infect them and reproduces inside this cell 30,000 times in a week. So 1,000 gives you, in less than a week, 30 million new parasites, which is a hell of a lot of parasites in your body. 1,000. And in Africa, most of the times the people get 18 infectious bites per night. Could you imagine that? Well, that gives you an idea why it is that there are so many. Not only the parasite reproduces 30,000 times per week per parasite, but also change completely the morphology. Now it is not like a larva-like structure, but a pear-like structure. We call it merozoites. These merozoites invade the red blood cells in less than 40 seconds. So you have to be very wise to stop an infection that takes less than 40 seconds. And you have to be, how I say, to know very well the mechanism behind. Each infection of each global red blood cell produces 32 to 50 new parasites every 2 days. So imagine that 50 times 50 times 50 times 50, and that's the reason there are so many cases of malaria and there are so many deaths of malaria as a consequence of that. So that this is the parasite in the merozoite, as we call it, or the pear-like structure, which is the merozoite. And among them, there are a few of them that become banana-like parasites. So that in essence, you have a larva, you have a pear, and then you have a banana. And then that's exactly the way it works. And these ones are the ones that are taken up by a non-infected mosquito, and then the cycle repeats over and over again. So in essence, you can stop the parasite at the larva-like structure or at the pear-like structure. Yeah. So these are the two ways and two probable methods to develop vaccines against malaria: stopping it at the parasite, stopping the parasite at the larva-like or at the pear-like structure. Everybody concentrated the efforts into this stage, but I decided to do it the other way around. It works very well. When one thing does not work that way, look at the reverse. It works very well. So that was the way I decided to work on the merozoite, meaning in other words. We had also another advantage, that it is that in the middle of the Amazon jungle, discovered by one of my mentors, Ronald Mackenzie and Carl Johnson, there is this monkey in the Amazon. This is the monkey, I have to say that, This monkey, which is the Otus monkey. It was named by Humboldt, Otus, because if you can see, it looks like he didn't have any ears, which in Greek means a without, Otus ears. That's the reason of the name of this monkey. And you can see that it has very big eyes. It's because it's a nocturnal monkey. This is extremely susceptible. That was discovered by Carl Johnson and Mackenzie. It's extremely susceptible to development of malaria, human malaria. It's the only species where you don't have to do anything to induce the disease. Just inject it, and then the animal develops the disease with the same characteristics as humans do. It turned out that years after, my son, who is Antonio Muro's colleague and partner also, decided to do the genomics of this monkey. And he found that I will show you later on, that at the immune system level, at the DNA level, this monkey is 95 to 100% identical to us. So we were very lucky because we have a very susceptible animal because of, immunological and also hematological reasons. They did the proteomics of the red blood cell of these monkeys, and they found that they share the same molecules that we humans have. So not only the immune system molecules, but also the red blood cell molecules are identical between the two of us. The monkey, and that's the reason I call this the picture of the two monkeys, so that's exactly the reason of that. So what happened, it was that with that, what we did, it was that we took and dissected the proteins of the Plasmodium falciparum parasite, pear-like, meaning in other words, the merozoite. Trial errors, looking at the amino acid sequencing, and not only that, but synthesizing the peptides according to that. We developed a methodology to vaccinate the monkeys, treat them, and then release them in the jungle later on without any other problems. That's the advantage of working with malaria, that you can treat the animals or the people, and then they get cured, and then you can back again, return them either to the jungle or to a place where you are working at. So that was what it happened. You know, trial, error. We found 31, 95% or 99% of the population in here, you know, had not been born by the time that we published that. Probably Ignacio and myself were by then around, you know, so that's the point. So we published by then the induction of protective immunity against experimental infection in the monkeys with 4 pieces of chemically synthesized peptides of the Plasmodium falciparum parasite. And that was the first chemically synthesized vaccine ever published, you know. And it protected 50% of the monkeys. And that was, well, with that molecule that it was called later on SPF 66, and here you have the amino acid sequencing, you know, we developed the first chemically synthesized vaccine that later on we tested in humans. Obviously very few at the beginning, and then later on, it turned out that we used it in thousands of people. So much that, for example, in here, we had 15,000 people vaccinated, and then later on, we went to vaccinate, we know, the Venezuelans vaccinated in Venezuela, the Ecuadorians in Ecuador, the Brazilians, the Tanzanians, and so on. So much that, as Esther just mentioned to you before, it was recognized by WHO, and we decided definitely to give that patent for free to humankind, period. I am not interested in money, you know. I need it only to do research, but that's all. Getting rich, there are so many rich around the world that I would have been a poor millionaire, you know, without that, with that. So that it didn't make any sense, really. So for that reason, we donated to the WHO the patent of the malaria vaccine. Well, the conclusion was that after being tested, that vaccine, the first one, remember that, the only chemically synthesized vaccine, in more than 50,000 people, the protective efficacy went from 20 to 40%. You know? So, to me, it was that definitely it could be used in large populations, because that meant, in other words, that you know, something like 60% of the population to 70% of the population were not going to be protected. So I stopped, and that was by 1996. It's going to be now 22 years ago. We stopped. Also, that there is a genetic control in the immune response, because the people who were black responded in a completely different way to the Indians and to the mestizos and to the Caucasians. So for that reason, there was that variability in the protective efficacy and in the immunogenicity. That was very clear to me that obviously there is a genetic control of the immune response, and that in this case, it was perfectly defined. But also, it allowed us to conclude that these molecules are pure. Here there are a lot of organic chemists, pure by HPLC, mass spec, and therefore they are devoid of any deleterious contaminant, because the molecules are purified. That's one point. They are easy to synthesize. This is a methodology that, just Esther mentioned to you before, developed by Bruce Merrifield. You know, they are practically cooking, absolutely. And they are reproducible from batch to batch, because the molecule has to have, you know, the same number of Daltons, identical from batch to batch. If not, you are dealing with another product. And also, they are stable at room temperature. This is a powder, you know, and then you can keep it at room temperature anywhere. And besides that, it is extremely cheap to produce. The new vaccine that I am going to show you today costs us 10 cents of euro. 10 cents. There is no vaccine that can beat that price, you know, and we are making a lot of profit with 10 cents. Absolutely, because you have to vaccinate 3.6 billion people twice. Sell it at 1 euro. So we cannot compete with Carlos Slim or Bill Gates, but anyhow, it's a good amount of money. That's the point. Then, based on that, we decided to develop the new vaccine. It was only 40%. The maximum was 40%, and then we decided, okay, we're going to stop in here because it needs more components. And the new vaccines that we developed, they will have the prefix Col, that means Colombian, and the vaccine Since we are now in the process of developing new vaccines against other diseases like tuberculosis, the one against tuberculosis will be called TUVAC, the one against dengue will be called DVAC, and so on. So what I'm going to share with you are the results of the called FAVAC, which allowed us to identify the rules for the development of the new vaccines. This was a consequence of the work by this student of mine, who's now professor at Boston University, Mauricio Calvo. Probably he had the nickname that probably could be applied to a lot of you. The nickname was Electron, because I couldn't find him. You know, wherever I was going to look for him, he was not in there, anyhow. And that's by definition the electron situation. So he asked me, okay, can I look at which ones of the molecules that you have synthesized and tested in the monkeys binds to the red blood cells? And I said, bingo, he discovered a way to make vaccines. Because rather than testing them into monkeys or humans, it was just an in vitro method, you know? Just looking at which ones of them bound with very high capacity to the red blood cells. And he found that the ones that are in red, which are the ones that were included in the original SPF 66, bound with a very high capacity to red blood cells. So for me, it was immediately easy to understand that what we were dealing with the first vaccine, it was in a receptor-ligand interaction. That's a very clear and simple concept, you know, where the receptor is present on the cells that are going to be infected, and the ligands are the fragments of the proteins that bind to the host cells with a very high affinity. If you are able to induce an immunity blocking this receptor-ligand interaction, you have obtained a vaccine. So in essence, the concept is extremely simple. You just block the receptor-ligand interaction, identify which ones are the ligands, and that was what we did. In essence, that it is the methodology. So by the time that we developed SPX66, there were only 4 proteins described, and that was the reason we were limited by the number of proteins. But then, later on, after 1994, and up to year 2000, lots of molecules were described. So what we did, it was that we synthesized, you know, all these molecules in pieces of 20 amino acids long, sequential. Yeah? And with the methodology developed in our institute, which is highly sensitive, robust, and not only that, but specific, because we made it totally specific, we were able to identify sequences that bound to the host cells with a very high capacity, the ones that are above the dotted line. But what it was important is there were some fragments where the amino acid sequence didn't change at all, which are the ones that are in green, while the other ones are variable. It is very well known that microbes escape the immune response by a variability in the amino acid sequencing. One single amino acid change in that molecule immediately allows the parasite to change the susceptibility, and not only that, to cheat the immune system. So for that reason, we dropped completely these variable sequences, because the malaria parasite has at around 200,000 variables. So 200,000 variables is a hell of a lot of peptides to synthesize. So we concentrated our efforts into the conserved ones, which are that one. So what we did, it was that we started looking at which ones were the binding sequences that are conserved. But since we want to go deeper than that, we synthesize analogs where we changed each one of the amino acids for a joker, which is glycine. And in this way, we were able to identify which ones were the critical binding residues, which ones are the susceptible to be modified and lose the capacity to bind to the host cell. So remember, conserve amino acid sequences and critical binding residues, because that it will be extremely relevant later on to develop chemically synthesized or vaccines, any matter, it doesn't matter which one. So in essence, that was, we were able to identify those conserved. And what we did, it was that we synthesized all the peptides that correspond to the different proteins in pieces of 20 and identify the black bars that are in the green areas or the red ones. I will talk to you about the red ones later on. And in essence, we synthesized them all. The 58 proteins that are involved in the invasion to the red blood cells were screened and analyzed in the way I mentioned to you before. Remember, conserved binding sequences and critical binding residues of the 58 relevant proteins that are involved in the invasion process. That gives you practically all the possibilities to develop the vaccine. So that was the point. These were not only binding, but also they performed some other critical biological functions. Some of those ones activate the calcium signal to invade the red blood cell. And also some of them have enzyme activity, serine proteases. Some of the ones have, for example, like HATPase, yeah? And some of the ones are growth factors, like epidermal growth factors. So not only we are blocking the invasion, but we are blocking the other biological functions that are relevant for the parasite to survive. So that we are killing the parasite at two different levels, and probably that it will happen with the other diseases. Not only the impeding the binding and the infection, but also impeding the biological functions that they are performing. All of this has been published, you can see that, long time before. Besides binding to the host cell receptors, conserved high-activity binding peptides perform some other relevant biological functions like the ones I mentioned to you before. We have published that, as Esther mentioned to you before, in the most relevant scientific journals, like, for example, Chemical Reviews, which is the number 4 out of the 18,000 journals that are published each month, number 4 or number 3, and these are the ones where we publish our data. And that's probably one of the problems we have, that we publish in chemical reviews, and our colleagues that are physicians or are biologists don't look at these journals. So we went to immunize monkeys with those chemically synthesized peptides, and that was a disaster, a real disaster, because we have not been able to produce one single antibody against the conserved binding sequences. But we are able to induce huge antibody titers against the variable ones, but they are specific against that variant, against that sequence. So therefore, you have to deal with 200,000 variants, and that it is an impossible task. Yeah? So we did that, and we were lost for 7 years, vaccinating monkeys and infecting them, and they got the disease full-blown, and treat monkeys just not to allow them to die. And then, being an immunologist by Conkel, I decided to add fragments that were associated with the helping in the immune response. They are called T helper epitopes. At the beginning, at the end, here, which are the ones in blue, at the beginning, at the edge, short, long, and so on. And we went to vaccinate monkeys, and it was equally disastrous, because what happens is that the immune system sees what you add, but not the binding sequences. Let's call the binding sequences the hands of the microbes, yeah? And that was very disastrous. These conserved high-activity binding peptides are immunologically silent. They are not seen. They do not suppress. No, they are not seen. It's absolutely, we're blind, completely blind to these amino acid sequences. The immune system don't see them. They just ignore them, and I will explain you later on why. Then it turned out that I said, okay, let's look at the critical binding residues. Let's modify them using, back again, a joker, which was a glycine. And we went to vaccinate with peptides where we had changed it already, replaced already the critical binding or the non-critical binding residue. Went to vaccinate monkeys. Bingo, that was the answer. You have to modify the critical binding residue. That's the trick. You have to identify it and modify it. Well, it turned out that each one of those ones, those residues, can be replaced by any one of the 20 amino acids. So we started this task, where we decided to look at which amino acid could replace to the other one, the critical binding. And we started that work where we synthesized 40,000 peptides, 40,000 peptides, and we tested 4,000 of them in the monkeys. And obviously, you can see negative results when some replaces were not the appropriate ones. Some of the ones induced antibodies, but no protection. I will explain that later on. And some of the ones induced antibodies and protection. So we can dissect the immune response very well. Immunogenicity and protectivity, which it is what one looks for. You know? So this is part of the different papers that we publish to identify. Let's go quickly, very quickly. You know, here you have just to reach, next please, yeah? Just to reach the conclusion, next please, just to show you that, next please, that you had to replace those critical binding residues for another one having the same mass, the same volume, the same charge, but opposite polarity. So that means that I can replace the thumb only by this thumb, but not by this small finger. And this small finger, I can replace it by another small finger, but not by the thumb. So in essence, what you have to do is to identify which one is the critical binding residue, and once you know what residue is that one, replace it by another amino acid with the same volume, charge, mass, but opposite polarity, in such a way that you have to replace, whenever you have a critical binding residue where you have alanine, you replace it by serine and vice versa. Proline, that has 113 cubic angstroms, by aspartic acid that has 111 cubic angstroms, and so on. This has been published long before, so that we now know that if you want to make a vaccine, identify the conserved binding residues, conserved amino acid sequences, the critical binding residues, and change it for these replacements. Now we can produce antibodies against those molecules that are not naturally seen, evolutively not seen by the immune system. So that's the reason these vaccines are not the classical vaccines. These are modified that go beyond evolution. And that's exactly what we are doing, identifying to modify according to the new rules. That these are the ones that I want to share with you. So in essence, that we induce antibodies that recognize by fluorescence, by Western blot, the recombinants, but the most particular important thing is that the animals do not develop once they are vaccinated, they do not develop one single parasite in their blood. Meaning, in other words, that we are inducing a sterile protective immunity. Sterile. No parasites in the blood at all. Obviously, that we have a hell of a lot of those proteins or those modified ones that induce protection, but partial protection for some time. And obviously, these are the ones that are not perfect, and therefore we discard them. Yeah? So that's the point. So that this is what we published a long time ago in chemical reviews. In conclusion, is that to render these peptides into highly immunogenic and protection-inducing, some critical binding residues have to be replaced by others with similar mass, volume, and surface, but opposite polarity. That it is the trick to develop vaccines. So these are some of the components of the new malaria vaccine, which my lawyers are very paranoid. They say, obviously, don't publish that, don't show it, because somebody can copy it. And I say, yes, they can copy this, but not the methodology and the results. So that's the point. So that's there. So we can produce antibodies against any one of the malaria parasites. So that we did, it was that we went backwards. We started working originally with the pear-like structure, and that was the way we identified the rules. But now we are working with the larva-like structure, and the same gets boring, because it's the same in the sense that you synthesize the peptides, identify which one of them bind to the hepatic cell, not to the red blood cell, or to the endothelial cells, and those ones are the ones that are modified later on, all of them, they are modified to induce a very good protection against the first stage, which is the larva-like structure. In essence, what we have is that we have vaccine that protects against both stages, which nobody has already achieved. You know, the people have already tried to develop against the larva-like, and some other ones have been trying to develop against the pear-like, but in essence, we have both, even the tree, because also we have produced antibodies against the banana-like structure so that we can protect not only the liver, the red blood cells, and the mosquito, which are called the altruistic vaccines, because it's to protect mosquitoes, not humans. You know, it is the way. So in here you have all that information that has been published long time before. So in essence, we have already developed the immune protection inducing protein structure concept, which is different from the classical vaccines, because these are natural molecules that are modified according to the rules I mentioned to you before in order to make them visible to the immune system. While in nature they are not seen, we convert them into very visible ones to be able to induce not only antibodies but also protection, which it is something that it has been impossible because the way most of the times the microbes evade the immune response is the mutation or the blindness. And that was one of the things that, thanks to the invitation by Antonio Muro, we were discussing last night with Philip Sharp, you know, because he's working on cancer, and I told him, listen, one of the problems with the cancer cells is that they are not visible to the immune system. It's the same problem that we have in here. And the way I sorted it out, it was this. He said, okay, let me invite you to give a talk in our institute. And I said, fantastic. But somebody did it before you, which was Harald Surhausen. Well, I was very happy with that, but not completely happy, because that tells you that you have to identify, but there are no rules behind that. So for that reason, I started working on the three-dimensional structure, and 25 years ago, thanks to the collaboration of this guy, by the way, I have to tell you that I'm very grateful with Toño by the invitation, but you know, the Universidad de Salamanca doesn't pay me the same fee that we charge. You know, we have this circus, as we call it, you know, in here just to give talks all over around the world, and I cannot tell you how much they pay us. But anyhow, well, it's the point. And then Kurt Buttrig was the one who discovered the nuclear magnetic resonance long before, and so we established our nuclear magnetic resonance 25 years ago, you know, with 600 megahertz and 500 megahertz instruments. And in such a way, we were able to determine the 3D structure of the molecules, identify exactly where it is that each atom is located. In the general protein, we know that our peptides are located in the places where they do interact with the receptor cells. So that it is absolutely true, you know. We have identified those ones. And if you look at that, look at this. This is something very beautiful. In blue and in yellow are the binding sequences, and look at the areas where it is red. In the protein, the complete protein, what it happens is that this is what it is visible to the immune system. And this is the area where the parasites mutate a lot. And we have already detected that, and that was the reason that paper was called Far from the Madding Crowd, meaning, in other words, that if this is the binding sequence, the hand, what the parasite allows you, or the microbes, because this we have done for hepatitis, for tuberculosis, for malaria, if these are the hands, what they allow you to see is here, 180 degrees, so that there is no possibility of the antibodies to block the binding. Because what the parasites, the microbes, show always are the red areas, which are here, so that it is what it happens. Meanwhile, the binding sequences are the interior part of the molecule, so they are not visible. That's one of the reasons they are not visible. You know, another example, just to give 3 out of the 25 examples that we have, back again, the same story holds true. You know, where you have in here the binding sequences, and look at the place where the exposed or variable or highly immunogenic areas are in red, very far away, as I mentioned to you before. Another example in here, these two are the ones that bind to the host cell And if you look at that, look at this yellow and blue, and look at the exposed areas to the immune system. Just exactly in the back, 180 degrees. And that allowed us to identify some of the rules for the immunological or the escape to the immune system reaction. In here you can see, obviously these are the antibodies. Look at what they do recognize. Look at the place where the binding sequences are present. Very far away, indeed, you can see that. These are in green, the antibodies, and these are the binding sequences, and so on, and on, and on, and on. Let's go quickly through this. In essence, what we have done is that we have determined the 3D structure of 400 binding sequences, native and modified. And that allowed us to compare between the native that are not seen by the immune system and the modified that are highly immunogenic and protection-inducing. So that's the reason we are very happy and content what we have achieved so far. So what happened was then that we shifted into the human immune system. Regarding this case, the human immune system, the ones that see the vaccines or the molecules are associated with the histocompatibility leukocyte antigens, which in this case, if this is the vaccine, it has to fit perfectly well into these molecules, which are the ones that are going to present the vaccine to the T-cell receptor. We will go through that very quickly. And then in here, the T-cell receptor interacts with this complex to induce the appropriate immune response. You can imagine that definitely it has to fit perfectly well. It has to fit. If not, you get a weird response. Therefore, you have to make your vaccine perfect. I will show you that you are not allowed to have more than 15 degrees out of 360 possibility of mistake or deviation. If we look at the genetics of the immune response, as I mentioned to you before, in here, these molecules are the ones that present the vaccine to activate the immune system. They are a tremendous variability. So for that reason, the people who are tackling the problem from the biological point of view, they have to face the genetic variability of the parasite, which we have already sorted out, and the genetic variability of the person who's going to receive the vaccine. So we wanted to solve that problem too, and that is what I want to share with you. So in this case, as I mentioned to you before, this monkey, Toño, you should have your picture in here. Okay, this is my son, okay? He comes very often to Salamanca. This is the monkey who decided to look at the immune system or the genomics of the monkey, and then he did all the cloning DNA sequencing of those molecules, and Manuel Alfonso is his name. He is now on sale, I want him to get married, There is the open offer, Okay, so Manuel Alfonso is the one who did that, and he was the one who found that the molecules are something between 90 to 100% identical. Most of them are in between 95 to 100%. There are very few ones that are below 95. But anyhow, one has to describe the lower limit. And then that was the point. If you look at the molecule that presents the vaccine, in this case, it happens that that molecule has a throw in here, or channel, where the vaccine has to fit. So from residue number 1, amino acid number 1, to amino acid number 9, there must be a 26.5 angstroms distance. But look at how wise are the microbes. The microbes, in essence, what happens is that they usually have molecules that are, look at this, only 16 angstrom or 17 angstrom. So that in essence, they are 10 angstrom shorter. And they cannot fit well at all, because being so short, they cannot fit perfectly well into the 26. But when we modify those molecules according to the rules I mentioned to you before, these molecules fit perfectly well into the pockets of these molecules. So therefore, the molecules can be presented, the vaccine can be presented in the appropriate manner to the immune system to activate the immune system. So this is pure genetic engineering and chemical engineering to develop vaccines. So the modified peptides, the ones that induce protective immunity, have 26, remember. They must have 26.5 angstroms. This is 26. Next, have 27. Next, have 26. Next, and so on. So in essence, to render a molecule highly immunogenic and protection-inducing must have 26 angstroms, and we have reduced all nowadays to 1.5 angstroms difference, so that it is very narrow to make it perfect, yeah? They must have, okay, now we are not talking about chemistry, we are not talking about molecules, we are talking now about physical measures, parameters. Now they must have a certain conformation, and in this case, the torsion angles. Look at that. Because if you look at this hand must have this thumb with the appropriate surface, the appropriate volume, but the fingers must have in this orientation. So that it is what it is called conformation. And in such a case, you have in here this angle of the peptide bond, you know, where the C must have something in between 100 degrees and the other one minus in the phi, yeah, minus 108. If you look at our molecules that are inducing protective immunity, you see minus, in this case, is minus 80, and in this other angle, you must have 120. So please, pay attention now. We are not talking about amino acid sequences. We are not talking about anything. We are talking now distance, torsion angles, and imagine that that can be reduced to mathematical rules. And that is our objective, to identify the mathematical rules to develop vaccines against any disease. And that it is the critical point. Now, if you look at that, based on that, we can induce antibodies, we can induce protection against anyone. And as I mentioned to you before, I have only 15 degrees of difference, yeah, to make the molecule perfect, yeah. But besides distance, torsion angles, steric and electronic principles, you know, back again. You must have certain charges in this case with some free electron pairs, as it happened in this situation, or with sigma bonds in this case, or with these P resonance structures. So these are charges, yeah, and these are universal charges. Those amino acids have that charge, and according to that, you can modify the molecule to make it distant, torsion, charge, and volume. That's the other parameter. And in here, those modifications allow the molecule to fit perfectly well into the molecules of the immune system. And that it is what I wanted. They form a four-like structure. This being the molecule of the immune system, this being the molecule of the vaccine, they have to interact in this portion, in this way. Opposite, because if they interact like this, it will be a completely, how will I say, weird reactivity, and there will not be any reaction, so that it is the point. So, we know that also. Yep. We know that thing. I have still 10 minutes, just there, isn't it? Therefore, they must have a particular structure, which that structure is named polyprolynyl 2 left-handed structure. So that it is distance, torsion angles, and the torsion angles must display a polyprolynyl type 2 structure. Remember that the proteins have alpha helical, they have beta sheet, they have beta turns, and these ones die like a so polyprolynyl structure, so that this one. If you have that, you have developed vaccines, definitely. But these are the distances, these are in here, you know, the angles, the torsion angles. The distance has to be per rotation, 9.12 angstroms, you know, and so on. These are mathematical rules, yeah? It doesn't matter if it is tuberculosis, AIDS, malaria, whatever. These are rules, mathematical rules, which was what we wanted to have. Volume and charge, obviously, is another of the rules. And that it is exactly the point, what we wanted. Now, we can also determine that via different methods, not only via these ones that are usually working with methodologies that use prediction-based structures, and also what we are doing is to analyze them by quantum chemistry. So in essence, nowadays, we are doing very little research, but let's say biological or experimental research. We have converted this into numbers, into mathematical rules. And therefore, that's the reason I wanted to share with you. You can develop any vaccine later on. These have been published already. So just follow Tonio's suggestions and depend on you. If you want to develop vaccines against leishmania, dengue, we are involved in tuberculosis, or any veterinarian vaccines. If I suggest you veterinarian vaccines, it's because money is there, period. It's so simple. Because we can donate, we are going to donate the new vaccines to humankind, the new ones that are for humans. We are going to donate them, but we have to charge. We have to charge the ones that are veterinarian diseases. We are not going to be that generous, that full, So that in essence, that's what we do nowadays. Here, based on, there are something like 25 papers published by our group about look at that, the mobility in here, not only of the residues, but also the differences on charges according to the replacement of that amino acid in that particular. But also, you have to do an analysis on the T-cell receptor, the ones. Look at this. These are charges. These are P interactions in here. You have that, and you have to keep that in mind to present it to the T-cell receptor. And then when you present it to the T-cell receptor, the amino acids that are pointing upwards, they must have a certain orientation. So please remember, distance, volume, charge, torsion, and orientation. It's very simple. These are mathematical rules. You can see that. If you have that, you can replace 4 amino acids where the rules are fixed by mathematical methods. And then you develop the vaccine. That's the point, So that it is what it happens. These are the, obviously, tetrahedrons. Don't worry about that. We did it already. So that we are going to give just the formulas. It's very simple. Apply them, But this was what we had to do just to identify those rules. Which ones are in here, the sigma, or which ones are the P, bonds, or whatever, And that gives you the appropriate torsion angles and the appropriate rotations and the free electron pairs, the charges, and the p orbitals, and so on. And that gives you the orientation, just to activate the whole immune system in order to render that highly immunogenic. We now know that, for example, the amino acid number 1 has to fit perfectly well into the HLA molecule. But the amino acid number 2 has to be charged and moving toward the right-hand side. And the amino acid number 3 has to be uncharged and toward the left-hand side, and so on, Then it's very simple. Number 4 has to go to the right-hand side and has to be charged. Number 5 has to be vertical and polar. Number 6 has to be polar and toward the left-hand side, and so on, on. So that you have for that mathematical rules, period. You can identify them. And that was what I always wanted. If you look at that, there are no molecules in here. There are only torsion angles and charges. That's all what you have to do to identify that. And that was exactly what we did to identify which ones are the molecules. So that it is what it induces memory. Because you can induce a protective immunity that is very short. By the way, let me tell you something that since we have for this, we need for this 40 peptides, 40 hands, modified hands, and we have more than 200, who can guess what is going to happen? We are going to donate the first one, or we are going to sell the second one, which is going to be sold to the army and to tourists. I was called by the president of IATA. Said, Dr. Patarroyo, I would like very much to negotiate the vaccine with you. And I said, which one? Obviously, he didn't know the joke, but the joke is real. So he said, for someone who goes to Africa for 1 month, 2 weeks on a safari, yeah, to charge $100. $100 for someone who is spending $10,000 at least during the safari. $100 means nothing. I said, consider it done. So we are going to sell the second one to finance the research, not only here in Salamanca, but in Colombia too. Okay, good. That it is the point, yeah? So we have that, the complete 3D structure of these interactions, and if some of the lateral chains are in a wrong orientation, they impede the complete activation of the immune system. Yeah? Therefore, those inappropriately modified peptides can induce non-responsiveness, interference, blocking, or suppression of the immune system. So that one has to be very careful defining this. And the mathematical rules are done, absolutely, so that's the point. Let's have a quick look at what it happens. When the molecules are native, the hands that are native, meaning in other words, the ones that the microbes use to bind to the host cells, you know, in here, they have very particular interactions. Look at this hand. This hand has 5 hydrogen bonds, so that this makes the molecule roll, and it has 17 out of the 27 that are required to activate the immune system. 17 angstrom, so that is 10 angstrom short. You know, it's 40% shorter, 40%. How can this fit into the immune system? There is no way. So we were able to identify which ones are the mechanism parasites evolved to evade the immune system. Look at this one. It has 3 hydrogen bonds inside that gives you roughly 17 angstroms again, and this other one has 2 hydrogen bonds of 1 PCH interaction. In this case, you can see 17 angstroms back again. Or in this case, you know, you have 1 hydrogen bond on 1 PNIH interaction, yeah, and all of that. But whenever we modify them to render them into highly immunogenic, let's go quickly through these, you know, what it happens is that it has only 1 hydrogen bond, and the distance it is the appropriate because we modified them properly. And that it is. In the next case, is the same. 1 hydrogen bond, 27 angstroms, and here 27 angstroms, no hydrogen bonds, no PCH interactions, and in this case, back again, 27, 0 interactions, so the molecule is absolutely designed to fit perfectly well into the immune system molecules. That it is what we have done. And when you look at the aortus and human class II molecules and compare the 3D structure, which it is, if you look at the amino acid sequencing based obviously on the DNA sequencing, if you look at that, these are the interactions between the peptides that are in here establishing the hydrogen bonds. And if you look at this in here, this is the hole where the amino acid number 4 has to fit. And it is in the human and it is in the aortus. It's identical. So that was our good luck, that we choose, or we were, how will I say, forced to work with the aortus monkey that is based on the DNA sequencing and the 3D structure of the molecules. We have determined the 3D structure of these molecules. We know that they are identical. Not only at the amino acid and DNA sequence, but the 3D structure level. So that means that whatever we obtain in the aortus monkey can practically extrapolate it right away into the human. And that was a very good luck, definitely, you know, because this monkey is almost identical, as I mentioned to you before, to the human situation. Back again, This is quickly, yes. Could you click in here, please? Here, click, 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 click in here. Yep. Anyhow. Could you click it in here? Okay, it doesn't matter. Let's go quickly through this, okay? In essence, despite the difference in the amino acid sequence, when you look at the 3D structure, next please, when you look at the 3D structure, both of them are almost identical. Look at this. This is the whole, this is the human, this is the autos. So let's go quickly through this because I want to finish very quickly. Because according to Antonio, we are invited to have lunch, but I will see how good is going to be the lunch according to the talk, so that was exactly. Let's go quickly through this. In essence, if you look at this 3D structure of the molecules of the human and you compare the 3D structure, not only the DNA and amino acid sequencing, but also the 3D structure of the molecules, so we feel very confident and very comfortable with the result that we are obtaining in the monkeys to extrapolate to the human situation. Quickly, let's go quickly. 1, 2, please, 3, 4, and next, and so on, so on. So on is just to make the story very short, So next, please, Luisito, yeah? Identical, you can see in here. Look at this. This is the pocket in the autos. This is the pocket in the human. So that they are almost identical, based obviously in the difference that exists between nuclear magnetic resonance and crystallography, which it is what we apply to that. Next, please. Yeah, Luisito. In essence, the INPIPs should display the appropriate distance, torsion angles, side chains, volume and charge, and orientation to properly fit into the T cell, into the immune system complex to activate the appropriate response. Okay, let's go into the essence of this. Based on that, we developed 1983, 5 years ago, we developed this vaccine, which is called Colfavac. Contains 24 fragments modified according to those rules. And in here you have, next please, this paper had been already submitted to publication originally to Nature, but they didn't understand it because the essence of this case, it is the protective efficacy. And they were saying, okay, you have been claiming that chemically synthesized vaccines could be developed and there is nothing new in that. And I say, yes, you're absolutely right, but you didn't pay attention to the efficacy, which it is the clue issue in this area. So that it is, and now we are submitting into another one. This it is the amino acid sequence of the modified, the antibody titers elicited by them. And in here at the sporozoite or at the merozoite level, so that we have it. And here the antibody responses and the number of monkeys that are fully protected, 0 parasites in their blood, 0. That it is our criteria and we don't give up on that. So that it is the point. In here the HLA-D are typification of the monkeys by DNA sequencing and so on. And this is the one that will appear this year, published so far, 78.2%. And that it is what it has been, what it was developed till 5 years ago. 5 years ago, our institute in the Amazon jungle was closed because of the animalist attacks, really. They said, okay, you are smuggling monkeys rather than killing monkeys or mishandling monkeys. They said, you are smuggling monkeys from Brazil, Peru, and who has ever been at the Amazon jungle in Colombia, crossing the street, just across the street, it is Brazil. Cross the street. That means 20 meters wide. You are in Brazil. So I was accused of smuggling monkeys from Brazil. So illegal trafficking of monkeys, Pablo Escobar and myself, period. So that's it, So that this was the reason we had to stop that. And now this is the one I want you to go quickly through this, called FAVAC 90, that we will publish next year, where we have in here the amino acid sequence. With 40 peptides, we can cover 100%. 40, no more than 40. And in here, please, one back, please. And in here, you have 49 peptides, and you are covering 97.8%. against the merozoite, meaning in other words, the larva-like. Don't copy the formulas, okay? Just go ahead. Next, please, Luisito. Ale, okay? I told you don't copy the formulas, Luis. Come on. You know, he's leaving it for his father, Julio, who's here. Okay, next, please. Oye, que la próxima, ¿eh? ¿En serio? Joder, vale, ya, la próxima. Ya. Luis, la próxima, obedezca. Hombre, hágale caso a su papá, ¿vale? Sí, ya vale. Así de sencillo, ¿sí? ¿Ya? And this is against the larva-like structures. So we have one at the merozoite and another one at the larva-like structure. We have another one also against the banana-like, obviously. You know, so we are protecting against all. In such a way, we are able to cover the whole world population with only 40 peptides at each level. The whole world, meaning other blacks, Amerindians, mestizos, Asian, Orientals, Jews, Caucasians, and so on, so that it is our goal. Yeah? So much that we will start vaccinating in Ghana by the end of the year or beginning of next. You know, so that is going to happen. And that was published by the Ghanaian people, was 2 years ago, and then reconfirmed about 1 year ago, yeah, so that we will start that. In essence, you can leave this one or your lucho, ¿vale? Because there is nothing relevant in there. So, in essence, what it happens it is that with this methodology, you can develop vaccines against any infectious disease. But the surprise for me was this, where, you know, next, please. Yeah, where this guy who happens to be a very close friend of us since 30 years ago, Harald Surhausen, who was the one who discovered the papillomavirus as the cervical cancer agent, went to my institute. Later on, he invited me to go to Heidelberg to give a talk. And then he told me, hey, listen, Manuel, you made it against cancer too. I said, what? Listen, I am fed up with the microbes. Don't involve me in cancer. He said, come on, did you read my Nobel lecture? I said, no. And he gave it to me. And his Nobel lecture says, by 2008, that slightly more than 20% of the global cancer burden can currently be linked to infectious agents like viruses, bacteria, and parasites. And if you have developed a methodology to develop vaccines against virus, bacteria, and parasites, you're developing a methodology to prevent 20% of the cancer. That was long before. But last year, he published this data where he establishes that, you know, in essence, 40% of human cancers are induced by virus, bacteria, or parasites. So in essence, this methodology is going to be used to produce vaccines against cancer. Two, not only the infectious diseases. Please remember, in 1 minute I finish. Remember that, in essence, distance, torsion angles, charge, volumes, and orientations. And these are mathematical rules that you can use for any disease, for any tuberculosis, hepatitis, leishmania, whatever, and even for cancer. That it is the point. You know, the minute that I'm missing, you know, is because Luis didn't understand. Okay, so that in essence we have 20 biologically developed vaccines, but with this methodology, we think that we can cover the 306 infectious diseases that humans can develop. So that it is the methodology of the immune concept. And this is a paper that we are going to publish next year, where we give them the structural, stereo, electronic rules for fully protective chemically synthesized vaccine development. I don't talk about malaria. This is any vaccine. And that was what I wanted to share with you. Thanks, obviously, to Antonio Muro, his group, and to this old friend of mine, Ignacio Verdugo. Yeah? So that it is what it is I wanted to share with you, where electronic, geometric, and topological rules must be followed for effective vaccine development, which confirms the immune protection inducing protein structure concepts. And one of the things I always wanted was to develop this in our countries, just to show the people that universal problems, we can solve them. Because there has been always a certain tendency to, it was Don Miguel who said, que inventen ellos. No, in this case, we are not accepting that sentence. We are reluctant to accept it. And that was the reason I decided to work only with Ibero-Americans, as in this case, and this is my group. And I want to be well recognized. Not me, I am here, but in essence, these are the people who have been working and developing the methodology for vaccine development. They are the ones who deserve the credits, and mainly this guy who's here. Next, please, who is Manuel Alfonso, my son, who has been with me since he's 12 years old. So, back again, reiterating my gratitude to Toño, to Ignacio, to organizing committee for these kind of invitations, and you for your patience, I have not seen sleeping anybody so far, so that it is something I would like very much. Thank you very much, Esther, for that very kind. Next.
Well, thank you very much for your excellent, amazing presentation. It's always a complete lesson in medicinal chemistry. And I don't know if you had time for one question, no? Because I don't know, only one question, I think. So questions for the Professor Patarroyo?
Any questions, please?
Yeah, Beatriz has one question.
Yes, please. If you did.
Thank you very much. It was absolutely fantastic. It was impressive. And I was very, very happy to listen to your talk. It was the first time I was listening to you, and I'm impressed. And louder, even louder? Okay, I'll try to do that. I was going to ask you about how you administer these peptides, and what is the amount you have to administer?
Manuel.
How to deliver the vaccine? Yeah, okay. That it is the part that we had just finished. It takes only 2 shots, which is very important. And in essence, having 40 or 90 peptides, 90, you need 2.5 milligrams in total. And what we are now doing with the people from the Basque University is to deliver it by microspheres. That it is. Because our dream is to have it in one single shot. So we did lots of work long before, about 15 years ago with them, Juan José Luis Pedras, where according to the cross-linking of the polyglycolic lactic acid, according to the microsphere releases the vaccine the first day or 60 days after. So our dream it is to, how will I say, give it in one single shot. But we have not done that, and that's the reason. No, orally no, because we always try to simulate the system that the infection uses to obviously infect the people, and the mosquito, you don't eat the mosquito, so that's the point. So that's the answer. So far, the 2 shots are spaced 20 days. And the immunity, we have already been able to identify that it lasts for at least 3 years in the monkeys. 3 years, at least, So that's very important. And when we speculate that could be 5 years, which if you have to give a boost 5 years after doesn't mean anything. So that's the point. But one of the important things is it's extremely cheap. It costs us 10 cents of a dollar. we are going to sell it for $1 and for 100 the other one, you know, just to make it. Yes, thank you. Another? That's it. Thank you.
So I think we finished for the morning, I think. Thank you to all of you.
Thank you very much indeed, Esther. Thank you. Thanks to all of you. Me mandó la